Hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're really glad you made time to dial in today. We will be talking about long-acting injectables with Jean-Baptiste Langlois and Thomas Ulrich today. So again, good afternoon, Thomas and Jean-Baptiste.
Hello, everyone. Good afternoon.
I want to introduce our guests, Jean-Baptiste and Thomas. So starting with Jean-Baptiste, he obtained his PhD in organic chemistry from the University of Geneva in Switzerland, where he worked on asymmetric catalysis in the group of Professor Alexakis before he moved to the Massachusetts Institutes of Technology in 2012 to perform a postdoc with Professor Buchwald. And then Jean-Baptiste came back to Switzerland in 2013 to start his career as a medicinal chemist at a startup company named PQR Therapeutics, focusing on the discovery of novel oncology drugs. He then eventually started at the Novartis Institutes for Biomedical Research in Basel, where he's currently a senior principal scientist and a project team leader. Over the past 12 years, Jean-Baptiste worked across several different disease areas, including autoimmune disorders, oncology, musculoskeletal diseases, and neuroscience, and contributed to the discovery of 2 clinical candidates. Jean-Baptiste also received the Young Investigator Award in Medicinal Chemistry from the French Medicinal Chemistry Society in 2022, and was one of the most meritorious runner-ups for the European Federation Medicinal Chemistry Prize for Young Medicinal Chemists in the industry in 2023. Thomas, he obtained his PhD from the University of Technology in Vienna in Austria, and in 1999 joined the NIH in Bethesda in Maryland as a postdoctoral fellow for 2 years working on the synthesis of opioid receptor ligands. He then started his industry career with Novartis in Vienna before moving to Switzerland in 2008, becoming a group leader and director in medicinal chemistry at Novartis. Thomas's teams have advanced several drug candidates into clinical trials to treat musculoskeletal diseases, in particular rare bone disorders, sarcopenia, and tendon injuries. Now he's leading a group that is engaged in hit and lead generation in the field of immunology. So real pleasure to have you both here today with us. So yeah, welcome again.
Thank you, Denise.
Thank you so much.
Yeah, and in the good old Drug Hunter tradition, we like to learn a little bit more about our speakers. And I wanted to ask both of you, what inspired you both to start a career in drug discovery? And yeah, we can maybe change orders. So whoever wants to answer first now, we'll switch in the next question.
So I'm going to go first then. When I was in high school, my favorite classes were biology and chemistry by far, and I was very curious about pathophysiology. So, like, what is the mechanism of a given disease, and how can one restore normal function to affected organs or cells? And then I started reading books about herbal medicines and alkaloids, and how to manipulate alkaloids and change them synthetically. And it was, I think, then when I decided to get a PhD or a degree in biosciences and then pursue a career in medicinal chemistry research. So I guess it all started in high school.
How about you, John?
Okay. So for me, I think it's when I was in undergraduate, as an undergraduate student, I was already passionate about organic chemistry, but did not really know what job I would do with this. And I attended once a talk, it was in 2005 or 2006, a talk from Laurent Hennequin, who was director of medicinal chemistry at the French site of AstraZeneca. And I found really this talk inspiring. At the academic labs that I was frequently working, they were talking about the next methodology to develop, the next total synthesis to crack, the amount of the number of papers they would have at the end of the year, and Laurent Hennequin was talking about saving lives. So that was like really, something that got me from day one, that I could use organic chemistry to actually solve public health issues, and that really was inspiring.
Amazing. Yeah, telling us like teachers are so important and people who inspire us to be something larger than ourselves, essentially. Yeah, great. maybe the next question, what's your favorite molecule and why? Probably some molecule that helped patients, I'm guessing.
So then I start this time, Thomas.
Sure.
So from the recent approval, there is one combo actually that really I find mind-blowing, which is Cobenfy. I think Drug Hunter has a nice summary about this this combo. It's just fantastic. Everything is fantastic. The story behind this. So the main active ingredient is a M1 M4 agonist, which actually would not be tolerated as such, and it was studied in the 90s and could not progress because of systemic toxicity. So they co-administer it with a pan muscarinic receptor antagonist to actually limit in the periphery the activation of these receptors, and then they could have a profound effect in the CNS. That's just fantastic. And if you look at the structure of the molecules, both of them, xanomeline, for instance, is having this 1,2,5 thiadiazole. It's just something that we would not have usually in our programs, and trospium chloride as well is really fascinating. So that's really a cool one. It's meant to be a legendary drug, I think.
Absolutely, yeah. That's a great answer. Thomas, how about you?
Yeah, it's actually funny, and I'm not making this up because Jean-Baptiste and I have not talked about this before the webinar. But my favorite, let's say, drug product is also a combination of two molecules. But in contrast to the drug that Jean-Baptiste mentioned, the drug combo I'm talking about is fairly old. So these are molecules called salmeterol and fluticasone propionate, and they are co-administered as an inhaled drug. And I have a personal relation to that because when I was a child, I was suffering from severe asthma, and the standard of treatment at that time was short-term relief by beta-2 adrenergic receptor agonists. Now, first the invention of a long-acting beta-2 agonist, but then combining it with a glucocorticoid in one inhaled device was a game changer for patients, and it was certainly life-modifying for me as a teenager. And I think what's more, that if you look at those two molecules, they also reflect the beauty of how medicinal chemists can actually turn endogenous receptor ligands into powerful drugs. And that's what I appreciate most about that business.
Awesome. Yeah, no, it's really a great personal story. Those are my favorite stories. To if you have like a personal connection to a drug, that's always really most powerful. What are you excited most about in drug discovery today? Maybe Thomas can start this time.
I'm certainly excited about the expansion of classical oral drug space towards what we now call the beyond rule of five molecules, like targeted protein degraders, orally bioavailable peptides, long-acting injectables, to stay with today's topic. I still find it intriguing that we're talking about something like beyond rule of five molecules. So I believe that when Chris Lipinski introduced the concept, it was meant as a probability guide to achieve good oral absorption when you stay in that space. But many people use it in a very dogmatic way and introduced filters and rules, and that picture has certainly changed. And I'm very happy about that, because the boundaries of both drug likeness but also druggability have moved so much in the 25 years of my career. And we know nowadays that for drugging particular targets, we need molecules that don't obey those very strict rules.
Yeah, no, that's great. We had a whole session about this at the most recent ACS, and yeah, very, very fascinating topics with like oral macrocycles now becoming orally bioavailable, right? This is crazy what development has happened in the past, decades. Jean-Baptiste, how about you? What are you most excited
Very,
about?
very similar answer. I mean,
Yeah.
really the extension of the boundaries of druggability with small molecules. I think, just as a reminder, when I started my career 12 years ago, we could see in the scientific newspapers and literature articles saying that it was the end of the era of small molecules and that biologics would take over and other modalities would take over. actually, it was a bit concerning. I remember a specific discussion on that topic with some peers. And over a decade, I think RLTs, degraders, and also beyond rule of 5 molecules have really extended the power of what we can drug with a small molecule. So that's really fantastic and super interesting to watch.
Yeah, 100% agree. And yeah, if you could give your younger self some career advice, maybe the younger folks in the audience that are just starting their careers, what would you say to them?
Very short and swear. From my side, speak less, listen more.
That's what I said when I give a drug under flash doctor.
So that's as simple as that.
Couldn't agree more. Thomas, how about you?
Yeah, it's not very different. Well, maybe to expand a bit on that. First, I'd say I wouldn't do anything differently going back in time. And I had the privilege to meet many really fantastic people and to learn from them. And this is what I would tell myself all over again. Never stop learning and improving yourself. But when we talk about learning, the first thought that we usually have is, we are scientists, and so we have to improve our knowledge in science and technology. And there are infinite resources to do that, right? Never ending. But I think what's also very important in an industrial career is that we learn from our role models how to interact with people, how to form teams, how to lead effective teams, how to do proper stakeholder management, how to defend yourself, how to basically lead difficult conversations or resolving conflict. And these are all things that we sometimes refer to as soft skills in a quite condescending way, but those are super important. And that's also a piece of advice I would give to a young chemist in whatever organization they work. Learn from your role models also on the behavioral side.
Totally. Yeah. Thank you for that advice, both of you. And yeah, last but not least, what do you like most about Drug Hunter? Maybe Thomas, you can go first.
So Drug Hunter is something quite unique in my opinion. It offers to professional medicinal chemists a great opportunity to access, I would say, distilled information about all latest developments in drug discovery and medicinal chemistry. The point is, you can't access that without having to search and read tons of materials. Now, using Drug Hunter, I can familiarize with a drug or a target family within minutes. And that's the key point here, saving time, getting quick access to information. And I also enjoy a lot how additional information, once you want to go deeper, is actually hyperlinked on the websites and then presented in a very concise and nice looking way. So absolutely top.
Thank you, Thomas. How about you, Jean-Baptiste? What do you like most about Drug Hunter?
I have always been a keen reader of the literature, and I used to love the literature meetings when I was a student to exchange with my peers there. But when I started my professional career, I realized that it was a bit difficult to find an echo. When you share a paper, it's not everyone that has time to go through it and really engage the discussion. So I think that with Drug Hunter summaries, we can really have a usable, shareable source of information that we can really use to socialize a topic and then engage into productive and interesting discussion. So that's really helpful in this sense as well, on top of everything else that many people have mentioned and notably Thomas, but I think this is something I like a lot.
Awesome. Yeah, no, thank you so much. Yeah, we hear a lot that the shareability, like, basically encouraging, starting a discussion about projects, maybe reviving some of those journal clubs that's being helpful. But yeah, thank you so much. Yeah, before we dive into the presentation, I wanted to show, one recent new feature that we introduced on the website. So quickly sharing my screen here. And I'd encourage people to go here to the resources tab. We now organized all of our calculators here in this little tab. So you can just click on our calculators. The popular PK calculator has found its new home here, essentially. We have a scaled clearance calculator that you can use to calculate from your in vitro data to in vivo data. So for example, here in microsomes, you can enter a microsomal clearance, a plasma protein binding, and you'll get a predicted hepatic clearance with the well-stirred model, as well as an extraction ratio. And kind of as the next step to that, we recently launched the estimated human dose calculator. Dose is just such a meaningful parameter that you can optimize your campaign on, rather than just potency or just PK properties. So it's a very nice measurable, kind of composite parameter, if you will. So what you can do here is you can enter your IC50 in the most reasonable assay that you're following, plasma protein binding in animal species, molecular weight, plasma protein binding in human, and I'm just entering some values here to give us a number. You put in your volume of distribution in the animal model, your oral bioavailability in the animal model, as well as, let's say, a predicted clearance. And then you can use that, maybe the predicted clearance that you get from our scaled clearance calculator. You can change the absorption rate here. The default is moderate. You can also change it to fast or to slow, and you can change the dosing regimen, and then you'll get calculated a number of parameters, including the predicted half-life, as well as the predicted dose for a 70 kilogram human. So here in this particular case, with all these parameters, it'd be about 46 milligram dose. And again, that's something that you can check for multiple compounds and then optimize for the lowest dose compound. And with that, I will unshare and give the stage to Thomas now, first, to talk to us about the principles of designing a long-acting injectable drug. And then I believe Jean-Baptiste will elaborate on a specific program later for the TAK1 inhibitor. Thomas, stage is yours.
Great. Thank you very much. Cool. So, yes, before Jean-Baptiste is going to guide you through our drug discovery story on long-acting injectable TAK1 inhibitors, it will be my pleasure to present you with a few guiding principles towards the smart design of such very interesting drugs. A very short disclaimer at the very beginning. So I'm going to talk a few principles of physical-chemical parameters that are basically exclusively referring to small molecules with a molecular weight cutoff of around 1,000 Dalton. And the reason being that peptides and biologics, such as proteins or nucleic acid-based drugs, often require very special formulation options, and that is not part of the scope of this talk. So long-acting drug products like implants and devices, but also injectable long-acting products such as hydrogels, suspensions, polymer-based particles have been around for a very long time, and they have been invented particularly for two reasons: patient compliance and safety. We should distinguish between systemic delivery and local delivery of such LAI drug products. Systemic delivery, whereupon a drug is administered to a compartment in the body that serves as a reservoir, as a depot from which the drug dissolves, is absorbed and distributed through the systemic circulation to reach its target. And a very common example for that is a subcutaneous depot or an intramuscular depot. And historical examples for such drug products have been applied to metabolic diseases, central nervous system disorders, more recently to viral infections. Local delivery, on the other hand, the drug is injected into a compartment which actually represents the desired site of action, so where the drug should exert its biological function, and only a small fraction, if possible, should enter the systemic circulation and be distributed throughout the body, primarily for safety reasons. And an example for that is an intra-articular depot, where the drug is injected into a joint to treat all sorts of inflammatory or degenerative diseases of joints or bone or tendon. What those LAI drug products all have in common is they should extend the drug release beyond conventional solution-based systems. So we have to achieve a scenario by which the dissolution rate or the liberation rate of the drug in the depot, which equals then the absorption rate into the compartment, is actually slower than the elimination rate from, e.g., the systemic circulation. And then we get these so-called flip-flop kinetics, and it is the absorption from the depot that is actually the only determinant factor that influences overall drug exposure. The common approach in the pharmaceutical industry has been to take an already optimized molecule, usually an oral molecule, and to find and tailor a long-acting formulation principle that fits to the drug. The problem is that an oral drug doesn't necessarily have to be a very good long-acting injectable drug. Usually, the search for a good formulation requires the development and also the laborious characterization of many different formulation candidates, and that's a lengthy process. What we tried in our research groups was to rather optimize the drugs for the compatibility and the suitability with a formulation principle that has already been pre-selected. So doing it basically the other way around. And that led to the rational design of LAI drug substances. And for those who are inclined to dig a bit deeper into the matter, my colleagues and I have recently published this textbook here, Biodegradable Long-Acting Injectables and Implants, and it's accessible via the Springer editorial services. Now it's worthwhile looking into actually how oral and injectable drug molecules are different. For that, it's important to take a glance at the target product profile. And you all know very well that an oral drug has to dissolve well in the gastrointestinal tract, so high aqueous solubility is of high importance. It also has to permeate through the gut wall, so then it needs high passive permeability. And here we are excluding any drug absorption that depends on active transport. But the oral drug also should escape as much as possible hepatic metabolism to be able to distribute to the target tissues in sufficient concentrations. So high metabolic stability is also a parameter here. A long-acting injectable drug, on the other hand, should slowly dissolve in the compartment where it has been injected to reach that very, very slow absorption process that I've mentioned before. And this usually comes with quite low aqueous solubility. And that's also the main difference in physical properties between an injectable, long-acting injectable, and an oral drug. But what's also equally important is that the drug shows good compatibility with the medium or the carrier that it is injected with. So what we're looking for is very, very high chemical stability, especially in excipients that are needed for administering the drug, but also for manufacturing the drug. So these are typical specific organic excipients. Looking at crystalline suspensions as a prime example, and Jean-Baptiste will also refer to that a bit later on, crystal particles obey a very simple physical principle of dissolution. The drug release only depends on the dissolution rate of those crystals. And the dissolution rate is a matter of the particle surface, if we want to put it in simpler terms, of the particle size, and the intrinsic solubility of the molecule. And the solubility is a matter of the crystal lattice energy and the lipophilicity. Now, these are three physical units of measurement that researchers can manipulate: the particle size, the crystal lattice, the lipophilicity. When we look at the particle size, when we have too large particles, we usually get issues with injectability, but also tolerability because of foreign body reactions. If our particles are too small, they will probably dissolve very fast, and we will not reach our target product profile of weeks and months of release. Most commonly in industrial applications, we're looking at nanoparticles with an average diameter of 0.1 to 1 micrometer and microparticles between 1 and 10 micrometers. Now, when we talk about the buildup of the crystal, you all know rigid and flat molecules, if they are paired with strong intermolecular bonds that form hydrogen hydrogen bonds, van der Waals bonds, or pi-pi stacking interactions, this will result in very high melting, strong crystals. What we usually measure is a differential scan calorimetry to capture both the melting point and the melting event enthalpy to characterize those molecules. And it goes without saying, lipophilicity, that's the limited ability to form hydrates in an aqueous environment. Here we are typically looking at nonpolar structural features, so-called carbon-rich substructures, a large number of aromatic rings. And here we commonly measure the distribution coefficient between octanol and water, so log P and log T, to characterize those molecules. Now I'm going to present you with a very interesting relationship. This is a comparison of 11 clinical and or marketed LAI drugs. And what they all have in common is very, very poor aqueous solubility that actually enables them to release the drug substance up to weeks and even months after subcutaneous or intramuscular injections. But the way these drugs, these molecules achieve very poor aqueous solubility is different. The y-axis here is the melting point. The x-axis is the calculated log P. And you can see that the physical profile of these compounds leading to very poor solubility is driven either by a very high log P or by a high melting point. And for most of these compounds, these two entities are anti-correlated. And when we look at the two compounds here on the far end, very extreme calculated lipophilicity, we can see why. This is paliperidone palmitate, and this is aripiprazole loroxyl. These are both fatty acid ester prodrugs, and they have these long, floppy fatty acid extensions that make them super, super lipophilic, but also very unlikely to form strong crystals. On the other hand, when we look at compounds like carbodegravir, those are high melting, 250 degrees. The log P is rather low. Still, the physical endpoint in terms of solubility is very similar for both compounds. Let me draw your attention to one particular drug that is an outlier here. It's loresivivint up here. It's a solid form with an extraordinarily high melting point, 360 degrees. It also has a fairly high log P. It's not as extreme as the fatty acid ester prodrugs, but it's still relatively high. Now, this leads to a reported release duration of up to 6 months after injection into joint tissues. It's an osteoarthritis drug. And that results in a clinical dosing interval of only 2 injections per year, which comes with a lot of patient compliance. I'm going to show you 2 exemplary marketed drugs that are also based on crystalline suspensions, an old one and a new one. The old one is paliperidone palmitate, which I mentioned before. It's this fatty acid ester prodrug of paliperidone. It's very, very lipophilic. It's very, very insoluble. And marketed as Invega Sustenna, it's one of the highest selling long-acting injectable drugs of all time. So huge, huge marketing success. The new drug is lenacapavir, and you can see this fascinating molecular architecture of the drug. Also on the very lipophilic side, I could not find, unfortunately, the melting point reported for that drug. So if anybody from Gilead is on the call, if you're happy to share the melting point of lenacapavir, I'd be super curious. So the point here is, when you look at these very different structures, is that medicinal chemists can build in all desirable key molecular features. They can build in lipophilicity, 3D shape, aromatic ring count. But what all that requires is an early investment in proper solid state characterization. So crystallinity, melting, polymorphism landscape, stability upon milling processes. And to that end, it's very important that the chemists in the research departments have a close interaction with the pharmaceutics development functions. Lipophilic prodrugs, as I have mentioned, represent a special case. There are crystal-based LAI depots of small molecules made prodrugs and met with very, very great clinical success. Historically, these have been corticosteroids such as methylbrenisolone acetate, which here has been esterified just with acetic acid at decarbon 21 alcohol here, and upon hitting the bloodstream, esterases will cleave the acetyl group and release the parent drug methylprednisolone. Antipsychotics and antiviral drugs have complemented that picture. And if you take that prodrug principle to a lipophilic extreme, like, as I mentioned, paliperidone palmitate, this palmitic acid actually renders the molecule so lipophilic and so insoluble that you even can't measure aqueous solubility anymore. It remains to be said that you don't necessarily need an alcohol in a drug to form such a prodrug. Fatty acid prodrug principles are also possible for non-alcohols. The marketed example is aripiprazole. It doesn't have an OH group, but it has this lactam here. So the medicinal chemistry workaround was to attach the fatty acid ester via an aminal bridge. And then this, of course, leads to a biphasic elimination process to release the prodrug. It's also possible to encapsulate a drug substance in depot carriers because not every molecule can be optimized towards well-behaved crystalline material. And that's where the molecular carrier comes into play. It will protect the molecule from fast dissolution and distribution. And it's the carrier, then not the drug substance, that takes over the responsibility of the long-acting principle. And there are lots of different applications. There are in situ gelling systems, there are oil or lipid-based solutions, there are liposomes. The most successful and most commonly used LAI technology for encapsulation in suspension principles are polymer-based microspheres. And the polymer of choice is polylactic glycolic acid, or PLGA, that has been used by many companies, including Novartis. Here you can see the microscopic image of a Novartis drug that has been encapsulated in such PLGA microspheres. And there are quite a few drugs that have undergone the same principle of encapsulation, such as risperidone or naltrexone or triamcinolone acetonide. And when you look at those three structures, they absolutely don't have any common structural elements. So there is no chemical design principle that underlies the carrier-based approach. You even have different ionization states. That's a basic drug. This one is zwitterionic. This is neutral. But what they all have in common is they have quite limited solubility in water. And this is important because then they will not easily diffuse out of their quite lipophilic polymer environment into the aqueous phase surrounding the particles. But what we do need is high solubility in organic excipients used in the manufacturing of the polymer, for instance, dichloromethane. And I should add only one principle, that is salts. Salts can be also used as long-acting injectables. Any ionizable drug molecule can, of course, form a salt with an acid or a base. And depending on the choice of the counterion, the physical chemical parameters, as well as the ADME properties, will change or can change quite dramatically. And a good example for that is the drug olanzapine pamoate. Olanzapine, it's an atypical neuroleptic drug. It's actually quite well behaved in terms of solubility as two ionizable centers. So it's doubly charged at physiological pH. But when you pair it with this acid here, pamoic acid, you actually get a salt that is totally insoluble and very highly crystalline and suitable for developing a microcrystal suspension that can be injected. So the caveat here is that these LAI salt forms are commonly not very easy to achieve. One needs to find a salt that is super stable, nicely crystalline, low solubility, and also we need to avoid the dissociation of the parent and the counterion once the salt hits an aqueous environment. And despite there has been a lot of investment in modeling that, to my knowledge, there is no reliable way yet to really accurately design the salt properties based on which counterion to use. So summarizing part one, LAI drugs have been met with great clinical success rates, particularly in psychiatric disorders and infectious diseases. And sometimes the LAI forms have been superior to the oral formulations of the respective drugs, mostly in terms of patient adherence. Medicinal chemists can design desirable physicochemical properties towards low solubility crystal suspensions, towards fatty acid derived products, or as I mentioned, high compatibility with depot carriers. You will see in Jean-Baptiste's talk that boosting up lipophilicity always comes with a steep price to pay. Having very highly melting crystals is a different strategy. I'm not going to steal Jean-Baptiste's thunder. He will explain that in detail to you. It remains to be said that in all those cases, regardless of the technology, high potency is super important to minimize both the injectable burden, but also to warrant the sufficient exposure over a lengthy treatment period. As you can imagine, a drug like lenacapavir with a once annual injection, if you want to cover an AUC efficacious thresholds for a year, you probably need a picomolar drug like lenacapavir. As part of the Novartis Biomedical Research portfolio, we were interested in those design principles, particularly for a program or a portfolio of regenerative medicines towards connective tissue damage repair in joints and tendons. And with that, I'm happy to hand over to Jean-Baptiste.
Thank you, Thomas. I will just share from my side. Okay, can you see the slide presentation mode?
Perfect. Yeah, thank you so
Okay,
much.
great. So thank you, Thomas. So the case study we would like to share with you deals with the development of a long-acting injectable drug for the treatment of osteoarthritis. So I'm pretty sure that all of you know osteoarthritis, but let me give you some facts. The disease is characterized by a slow erosion of cartilage, which is referred to as catabolism, inflammation of the synovium, as well as thickening of the subchondral bone here. So the disease really affects patient mobility and with time their overall health. So the affected population is absolutely enormous. It's a gigantic number of 500 million of patients worldwide. Just to give you an idea, it would be like if more than 50 cities of the size of New York would actually be affected. It's just crazy. And despite this, there is no disease-modifying treatment on the market, making OA one of the most prominent unmet medical needs. I believe that the situation is also compounded by the fact that clinical candidates targeting catabolism, pain, or inflammation only have met with limited success so far. So looking for interesting new biological targets for the treatment of OA, we were looking for pleiotropic effects in order to have an effect on different aspects of the disease at the same time. And in this context, TAK1 was actually reported in the literature as a very interesting and promising target owing to its implication in the NF-kappa B and MAPK pathways, which were respectively controlling inflammation and catabolism in OA. Unfortunately, TAK1 is also involved in many very important biological processes, such as cell survival, tissue homeostasis, and cell survival as well. So inhibiting TAK1 systemically would actually lead to side effects that would not be compatible with the treatment of a chronic but non-life-threatening disease. So that's the reason why we consider the development of a long-acting injectable drug, which would be directly injected in the affected knee of OA patients. Intra-articular injections, I have to say, are actually very, very common procedures, which are used in millions of patients every year, and for which many formulation technologies have been developed. As Thomas, I think, nicely mentioned during his part of the talk, all those formulation technologies rely on the injection of an insoluble drug depot in the joint, which would slowly release the drug over time, maximizing the local concentration while keeping the systemic or circulating concentration to homeopathic levels. And to be a little bit more concrete about this, the soluble drug would actually be released or expelled from the joint within hours, while hydrogels or even better microcrystalline particles could stay up for months. And again, as part of our program, we looked into long-acting injectable drugs, small molecules formulated as such for the treatment of OA, and identified loresivivint that Thomas has talked about. And really the two key criteria or physicochemical parameters of this molecule, the high melting point and the high lipophilicity, are rendering low recidivins microcrystalline particles virtually insoluble in synovial fluid. And as Thomas said, only 2 injections per year are necessary to maintain therapeutic level in the joint. So we believe that based on this knowledge, we could engineer those physicochemical properties by design into a TAK1 inhibitor to similarly achieve a long residence time in the joint. So we started our program with mining of the Novartis collection and quickly identified compound 1 as a potent and insoluble TAK1 inhibitor, which was originating from a former IRAK program. So as said, the molecule was already active in a biochemical and cellular assay against the target, had reasonable selectivity against IRAK1 and 4. And the reason why I mentioned this is that we paid particular attention to the selectivity against IRAKs, owing to their involvement in the same pathway than TAK1, but leading to a different cytokine response. So if we want to study the role of TAK1 in OA, we need a selective compound overall, but especially against IRAKs. In terms of physicochemical properties, the molecule was, of course, not as extreme as loricivivint, but still had a high crystallinity with a melting point of 248 degrees, a medium-high log P of 3.8, and already single-digit micromolar solubility in simulated synovial fluid. So what we did was to actually prepare a binding model of compound 1 based on the co-crystal structure of a close analog with TAK1, which unveiled this interesting binding conformation. So the cyclohexanol ring sits in a chair conformation with an axial CO bond here to allow for an intramolecular H bond interaction between one NH bond of the picolinamide ring or picolinamide moiety and the oxygen of the cyclohexanol. And this key interaction confers to the ligand a pseudo microcyclic conformation, which then occupies a large portion of the pocket. On top of this, it made 2 direct interactions with the protein, one with the hinge from the isoxazole and one with aspartic acid 175, with the second NH bond of the picolinamide moiety. So at this stage of the program, we decided to prepare a small library of close analogs of compound 1, ideally with higher crystallinity and lower solubility in simulated synovial fluid, which we can advance in vivo to evaluate the influence of those parameters on intra-articular PK properties. And to realize this, we identified the position 6 of the bottom pyridine ring as ideally suited for a library purpose, solvent exposed, amenable to a late stage modification. The first group that we added was a methylamine, very simple, which increased the potency on target and the melting point to 264 degrees while reducing the selectivity on IRAKs. We then replaced the nitrogen by an oxygen, but unfortunately lost 40 degrees of melting point, so we excluded ethers early on. And then we added bigger groups and notably identified compound 4, which had an additional dimethylamide moiety here, which had an interesting profile, restoring the level of potency and selectivity of compound 1 while having a melting point of 269 degrees, moderate log P of 3, and low solubility in simulated synovial fluid. So we decided to take compound 1, 2, and 4, formulate them as microcrystalline suspension, and inject them into the knee of naive rats to evaluate their intra-articular PK properties. Given our interest in sustained exposures, we looked into the concentration, total and unbound, in synovial fluid 1 week after a single intra-articular injection. Starting with compound 1, we could see that the concentration after 1 week was really low and even negligible if we consider unbound concentration, suggesting that really the compound did not have the suitable physicochemical properties for the sought-after indication. Interestingly, compound 2 with a higher melting point led to much higher total concentration in the joint. Unfortunately, the high lipophilicity translated into high protein binding in synovial fluid and hence low unbound concentration. And finally, compound 4 with a similar melting point on compound 2, but lower lipophilicity, had a good compromise and high concentration, total and unbound, in synovial fluid. So for the sake of time, I really had selected examples here, but at this stage of the program, we had a limited data set, but enough to do preliminary trend analysis. And we were pleased to see that the concentration in synovial fluid 1 week after a single injection was proportional to the melting point of the compounds and inversely proportional to the solubility in simulated synovial fluid, which was kind of comforting us, supporting our optimization strategy. So keeping this in mind, we selected compound 4 and advanced it for further optimization, notably for its selectivity against IRAX. We did again a binding model here for compound 4 and looked at the constitutional sequence of amino acids in the ATP binding site of TAK1, IRAK1, and IRAK4, and looked for potential differences. And only 2 actually were observed and are highlighted on this slide. The first one is asparagine 114, which is replaced by an aspartic acid in IRAK1 and IRAK4. So we hypothesized that IRAX with a carboxylate in this position could do a direct or indirect interaction with the aminopyridine motif of compound 4. So what we did was to design a truncated version of compound 4 where the dimethylamide would be directly attached to the pyridine, which resulted in compound 5. The molecule retained the potency on target, reduced a bit the melting point, but increased the selectivity against IRAX to 50-fold. The second difference is the cysteine 174, which is replaced by a glycine in IRAK1 and a serine in IRAK4. This amino acid residue sits in very close proximity to this key intramolecular edge-bond interaction that governs the binding conformation and the rigidity of the ligand. So we decided to release a bit of this conformational strain, hoping that the IRAX would tolerate a bit less this extra flexibility. And we did that in a subtle way by removing the nitrogen of the picolinamide ring here and the second nitrogen on the other side. This led to compound 6, which had actually an even higher potency on target and really good selectivity against IRAX, unfortunately at the expense of the melting point, which also resulted in an increase of solubility in simulated synovial fluid. So then we re-engineered a bit of rigidity in our molecules by cyclizing the dimethylamide moiety onto the adjacent aromatic ring and identified compound 7, which really had an excellent profile. Good potency on target, good selectivity against IRAX, good melting point of 281 degrees, moderate low log P, and low solubility in simulated synovial fluid. So compound 7 was renamed TAK756 and advanced for further profiling. First of all, the compound showed good functional activity in human-derived cells. Notably, it showed that it could potentially inhibit the production of matrix metalloproteinase 3 as a marker of catabolism, interleukin-6 as a pro-inflammatory marker, and prostaglandin E as a marker of pain. So that would be really the pleiotropic effect we would like to achieve. We then looked into the kinome selectivity in a panel of 330 different assays where the compound was inhibiting only 20 kinases by 50% or more at 1 micromolar concentration, which is a promising selectivity score of 6%, irrespectively of the kinase expression in the targeted tissue. So that's, I think, fair to mention that among the 20 kinases, a quarter of them are actually not expressed in human joint tissues. So in vivo, the selectivity would be even higher. Still, the molecule was having some residual activity against kinases that are related to anti-inflammatory activity. So we wanted to make sure that our compound was active as a TAK1 inhibitor in cells. So what we did was to look at the downstream substrate of TAK1, NF-kappa B, and could demonstrate in synovial cell lines that TAK756 was dose-dependently inhibiting its phosphorylation, suggesting that our compound was really acting as a TAK pathway inhibitor. We then formulated the compound as microcrystalline suspension, injected it in the knee of naive rats, And we are pleased to see that a low dose, as low as 1/4 of a milligram, was sufficient to achieve high and sustained exposure in all tissues of the joint, while keeping the plasma exposure below 1 nanomolar at all time points of the study. So after a short ramp-up phase, the release profile of this molecule is really flat, achieving a 10 micromolar total concentration in synovial fluid, and even higher concentrations in other tissues of the joint, like the tibial plateau and synovial membrane. With this, we advanced the compound into a PKPD model, a rat joint inflammation model, which consisted in the injection intra-articularly of lipopolysaccharides and monosodium urate to trigger a burst of inflammation in the knee of naive rats. The molecule would be injected prophylactically 3 days before the injection of LPS and MSU to evaluate its ability to prevent the burst of inflammation. And we would monitor this by measuring PK and cytokine levels at their peak 6 hours after the trigger, and gene expression in meniscus. The molecule was tested at different doses, again, one single injection every time at different doses, which led to dose linear increase of exposure, except at the highest dose where we saw a bit of saturation. In this plot, we have unbound and total concentration in synovial fluid. Looking at CXCL1 as a pro-inflammatory marker, we could see an effect at all doses tested and a full effect at doses above 30 microgram. This was correlated with dose-dependent reduction of gene expression, not only in CXCL1, but also IL-6 and MMP3. The effect is a bit less impressive here due to more intersubject variability, but still statistically significant at doses above 15 microgram. So a single PKPD study is not sufficient to enable a robust PKPD relationship, but still we were interested to see that an effect was observed when the unbound concentration in synovial fluid was superior to the IC50 value in the TAK1 cellular assay, and a full effect was observed when we were above the IC90 value. So to conclude on this part of the talk, we have identified key physicochemical parameters to optimize intra-articular small molecule drugs, and notably lipophilicity, melting point, and solubility in simulated synovial fluid. And using those, we have identified TAK756, which stands out as a potent, reasonably selective compound with exceptionally good intra-articular PK properties, which will be used to further explore the biology of TAK1 in the context of OA. I would like to thank all the colleagues who worked with us on this program, and also the colleagues from the Novartis drug delivery community for their expertise and very interesting discussions. Of course, you, Denise, and the Drug Hunter team for the invitation and the opportunity, and everyone who attended this presentation. Thank you very much.
Yeah, thank you so much, Thomas and Jean-Baptiste. That was really, really impressive. Yeah, I learned so much during this talk, and we already have a lot of questions here as well, and I have some questions of my own too, but maybe let's start with some of the audience questions. First question is for Thomas from Yoshi. In the LAI salt form, you mentioned that you need to avoid dissociation of the parent and the counter ion. How do you assess that preclinically? Is this maybe measured as by the rate of counter ion exchange on a resin, or is there any other technology that you use?
I think that's a common way that in the pharmaceutics department they actually do that. I haven't seen it myself, but I assume indeed that is the case. So when we have these investigational salts, we try to look at the ion exchange rates in pharmaceutical test systems. I believe that's the case. Thanks for the question.
Awesome. Yeah, sounds good. And then another question from Yoshi, this time for Jean-Baptiste. In the drug discovery project, when did you take the decision of choosing the long-acting injectable over the oral dosing? He says, I'm wondering if the LAI is preferred in a particular indication, or if you do choose an LAI option based on the compound properties, kind of elaborating on what Thomas said at the beginning. Yeah.
A very good question, obviously. Actually, we did it at the very beginning. So we started with the aim to develop a long-acting injectable. And the reason is that the treatment of OA is a chronic treatment. And TAK1 is really associated with known side effects, which would be okay if you have a really acute treatment for some indications, but for osteoarthritis, we knew that we would have to treat patients for, chronically, let's say. At best, we could make enough benefits to the patient so that they can stop treatment, be off treatment. But chronic treatment, systemic toxicity on target, we had to start with LAI approaches.
Makes total sense, for sure. Then the question about the melting point experiments, do you see degradation as part of the melting experiment? John is wondering if you use DSC for measuring the melting point, and if that can discriminate between a phase transition and a degradation.
Yeah, I think it can. Jean-Baptiste, you can specify for the compounds that you have dealt with, but essentially we can weed out compounds that show in the differential calorimetry scan, let's say, complicated behavior such as degradation events, thermal rearrangements, multiple recrystallization, polymorphism. We usually did well by selecting compounds that had a very nice single clean melting point.
But yes, absolutely. I mean, there is really a lot of information you can get from a DSC. So we were using that for each and every compound. For this program, we crystallized several hundreds of molecules, like at least 300 minimum. And we could learn really a lot. We could see vitreous transitions. You could see if there is residual solvent. You can see if there is polymorphs. You can learn from the enthalpy of fusion, which is also an indication of the quality of your crystals or the robustness of your crystals. And you can differentiate a total endotherm and a complete degradation of the compounds. That is really something that you can determine. Yeah.
Awesome. Perfect. Yeah. And then a question I had myself, I guess for you, Jean-Baptiste, as kind of a medicinal chemistry question, the two obvious questions, I think. Have you ever tried the macrocycle? You mentioned that intramolecular interaction. So have you ever tried to lock in this conformation is the first question. And then the second question, you mentioned that difference with the IRAK1 and IRAK4 is that cysteine. So the next obvious question is, of course, have you ever tried to leverage a covalent compound on the project?
The microcyclic idea, we debated about this in the team. I remember we tried also, we had several series and we tried in another series, not in this one. We did not try because we were a bit afraid to lose 2 H-bond donors in the process, which were important for the overall profile of our molecule. So that's why this was not the approach we took. I believe retrospectively that this would have been quite interesting, notably on the selectivity profile, because we would have had really a rigid microcycle, which would have been very interesting. For the second question, which is the covalent inhibition, we always discussed about this with colleagues and decided not to pursue this. The reason is that every molecule of drug that is dissolved in synovial fluid stays in synovial fluid for 3 to 6 hours before synovial fluid itself is replenished. So basically engaging the target covalently would not necessarily help us more than having a sustained exposure, because we cannot really, in this case, deconvolute PK from PD. I think it was really discussed strategically. Ultimately, we should have probably tested this and did not, but we decided to be very focused, very streamlining our efforts towards the reversible compounds which we had.
Yeah, makes sense. And you discovered
working compound. As you know, there are actually covalent inhibitors for TAK1 that are reported in literature. So we knew it could have been a possibility. Yeah.
Awesome. Yeah, few more questions, I guess, for Thomas here. Again, about maybe the preference of an LAI versus a once oral dosing, and then the other aspect is the safety. We were talking about drugs that maybe are administered once a year or twice a year. How is the perception of safety if there's maybe a DDI or a safety event? you cannot really remove an LAI.
And I think that is very much to the point of the beauty or the advantage of an oral drug or a drug that you can take on a frequent basis, that you can stop administration at practically any time. With a depot, with a long-lasting depot, you can't do that, obviously. So, if I had a choice between a safe oral drug that has been taken once, and then an injectable drug, providing that they have the exactly same efficacy and safety profile, I would prefer the oral drug. For that safety perspective, of course, you have to submit your treatment principle to chronic safety monitoring, and look at of course the relationship between systemic exposure on one side, systemic exposure, and any potential side effects that you see in preclinical models. On the other hand, you would have to look at the local tolerability of the doses that you want to inject. So yes, there are certain safety considerations with local injectables or long-acting injectables that cannot be ignored.
Well, thank you. And yeah, I know we're already a little bit of overtime, but maybe let's end on one last question if you guys still have a little bit of time, and that would be for Jean-Baptiste. Jean-Baptiste, what's the main clearance pathway for the TAK1 compound, and which in vitro assay did you use to measure that?
So, yeah, several layers of answer to this. So we use the typical liver macrosome clearance assays in vitro. We had debates with our PK expert, and the focus at this stage of the project was really to make sure we could slow down the dissolution, to make sure that what reaches the circulation, the systemic circulation, is really homeopathic levels. And then we would care a bit less about the clearance mechanism, because we have anyway so low amount in the systemic circulation that even long-lived molecule would not be an issue. It would be excreted in the end. So that's actually what we achieved with TAK-756. The molecule in vitro is relatively stable. It has low in vitro clearance in rats. But in vivo, we have less than 1 nanomolar total exposure in plasma at all time points, even during the peak, because you can have a bit of the drug that is actually soluble in your formulation. So when you inject, you could have a little burst of release that goes into the systemic circulation. That would be the equivalent of your Cmax, if you want. And even there, we were below 1 nanomolar. So that's really how we solved this. So I would say excretion in the end, direct, touched.
Awesome. Yeah, thank you both so much again, for a very, very nice presentation. Again, I learned so much. Thank you for sticking around to answer all the audience questions. And yeah, thanks to our audience members for the interesting questions and the interesting discussion. Yeah, and we will be back here next week with Larry Hammond, this time talking about sequence selective translation inhibitors, as we were just talking in the beginning about new therapeutic modalities. And, this is a new small molecule therapeutic modality that Larry will talk to us about next week. With that, Jean-Baptiste and Thomas, it was a pleasure to be on stage here with you. And yeah, hope to see you again live soon.
Thank you very much.
Thank you so much.
All right. Take care, everyone. Bye-bye.